guys. For those of you that don't know me, I'm Kelly Barnett, and I'm here to talk to you about the second stage of protein synthesis, which is translation. So the second stage of protein synthesis is translation, and translation is when the information in the mRNA is actually read to assemble a protein. First, amino acids will be brought in and positioned in the correct sequence, and then they're joined together to form a protein. Remember, this is picking up from the third step of transcription that we talked about last class. So we've talked before about DNA replication. Remember, that's a process where DNA copies itself into new DNA that happens inside the nucleus. Yesterday, or last class, we talked about transcription, which is where we took the DNA information, we used a sense strand, which is 1/2 of the DNA molecule, to create our mRNA. And now we're moving on to translation, where we're going to take this mRNA to the ribosome, bring in amino acids, and create an actual protein. So what's going to happen is the 5' end of the mRNA molecule is going to combine with the ribosome. Now the ribosome is already made up of protein and the RNA called rRNA, which is ribosomal RNA. That ribosome is going to move along the mRNA molecule in a 5' to 3' direction, reading one codon at a time. If you remember last class, we mentioned that a codon is 3 nucleotide bases on the mRNA strand, so 3 letters at a time. So in your notes on figure 6, the first codon at the 5' end is read as AUG rather than GUA. Remember, mRNA is approximately 1,000 or more bases long. It's read in those groups of 3, which make a codon. The codon is the secret code word for an amino acid. So here's the example we're talking about. Here's your ribosome, the big brown molecule. The purple here is your mRNA. So we're going to read this as AUG. That's a codon. Here is a picture of the translation process that's incorporating the 20 different amino acids. Remember, it's the sequence and number of those amino acids that's going to determine what kind of protein you're building. So here again, you've got your ribosome, you've got your mRNA, your 3 bases that make a codon, and then these things here are tRNA, and they're going to actually carry the amino acids. Whoops. So ribosomal RNA, I mentioned that just a second ago. It's rRNA. It consists of 1 large and 1 small subunit for each ribosome. The ribosome also contains proteins, and it's those 2 subunits that are going to hold the mRNA and tRNA in the right positions so that the amino acids can be joined together. Oftentimes you have several ribosomes moving along that single strand of mRNA. So again, here's your small subunit of the ribosome, and the large, they're holding the mRNA and the tRNA molecule in place. As the ribosome is moving down the mRNA molecule, a third type of RNA called transfer RNA, or tRNA, is going to bring specific amino acids to the mRNA ribosome complex so that they can be joined together. Although the tRNA molecule is single-stranded, like all types of RNA, it is folded back on itself, and it resembles a clover leaf shape. So if you use your imagination, you can see that it kind of looks like a clover. It's green in this picture to help you visualize that. It's going to hold an amino acid on one end, and it's going to hold what's called an anticodon on the other end. So here's that same picture. Imagine the clover, here's your anticodon, and here's your amino acid up at the top. During protein construction, the transfer RNA or the tRNA will actually transfer the amino acids to the ribosome. So you can think of transfer RNA as being a taxi that's moving the amino acid molecules. So transfer RNA acts as a taxi for amino acids. It picks them up in the cytoplasm and brings them to the ribosome where the proteins are actually made. At the 3' end of the tRNA molecule is an area that we call the amino acid attachment site. That's where your different amino acids actually attach and bind. Every tRNA molecule is specific and it can only carry one particular type of amino acid. So this tRNA molecule here can only carry lysine, for example. That's how that would work. Here you can see your mRNA strand is moving through the ribosome. You've got multiple tRNAs coming in, forming bonds. Here comes another one moving in to join up. So at the base of each tRNA molecule in the bottom loop is a special sequence of 3 nucleotides that we refer to as an anticodon. These anticodons of tRNA are complementary to those codons in the mRNA. So it's 3 letters that pair up with the mRNA. When the tRNA brings the amino acid to the ribosome, it gets positioned in the proper place or sequence by matching its anticodon with the codon of the messenger RNA. Then it will drop off its amino acid. The tRNAs are released back out, where they can combine to another molecule of the same amino acid and repeat that process anytime that code comes up again. As your amino acids come in, they're going to be linked together by peptide bonds, and then that chain of amino acids forms what we call a polypeptide chain. And it's going to continue adding on to that chain until they reach a termination codon. Termination codon tells you, stop, we're done building the polypeptide, and it's not going to read any other letters past that termination codon. If your polypeptide chain is functional, it forms a protein. However, many times it's going to take 2 to 4 polypeptide chains to actually form a functional protein. So here you can see the transcription of mRNA from DNA happens in the nucleus, moves out to the ribosome, and it attaches. Then the tRNA, here we've got the GGG codon of mRNA. It pairs up with the CCC anticodon of tRNA, which happens to carry the amino acid glycine. AAU codon would pair with the UUA anticodon, and it carries asparagine. And it continues on like that. Notice between each of these amino acids, you have a bond. That's a polypeptide bond. And this whole thing will be your polypeptide chain, which could equal a protein. This is just another picture of the same process. Okay, this one is just showing you again, we had transcription happening inside the nucleus. Our DNA opens up. We read off our sense strand to form mRNA. It travels out a nuclear pore to a ribosome. Remember, the ribosome contains our RNA. We have our large and small subunit holding our mRNA in place. And then you've got your tRNA molecules that pair their anticodons with the codons of mRNA. And a new one would come in, they would form a bond between the amino acids, and eventually you'd get your polypeptide chain or your protein. These are the 3 types of RNA that we find in our bodies. You've got messenger RNA, remember that just looks like a little string. Ribosomal RNA is inside the ribosome, and then your transfer RNA is the one that we said looked like a cloverleaf shape with an amino acid attachment site and the anticodons. Okay, so that's it for translation today. Just remember, transcription starts with a trans and then a C. That's the first stage of protein synthesis. And then the second stage is the translation, trans and then the L in the alphabet. It comes second. So hopefully you've learned something today. We're going to move on, and I'm going to explain what you're supposed to do with your worksheet.